Hello everyone, welcome to today's live broadcast, Sample Preparation and Analysis Workflows for Released N-Glycan Analysis of Biotherapeutics. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by LCGC and Biopharm International and sponsored by Prozyme. Founded in 1990, Prozyme Incorporated is based in Hayward, California, and offers products and services to customers at biotechnology, biopharmaceutical, and academic institutions across the world. Prozyme's flagship product line is an extensive glycobiology reagent portfolio alongside streptavidin, phycobiliprotein, and conjugate offerings. For more information about Prozyme, please visit prozyme.com. Prozyme is a part of Agilent Technologies, which is a global leader in life sciences, diagnostics, and applied chemical markets. With more than 50 years of insight and innovation, Agilent instruments, software, services, solutions, and people provide trusted answers to customers' most challenging questions. Information about Agilent is available at agilent.com. Before we get started today, we have a few housekeeping announcements. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. And if you have any technical problems viewing or hearing the presentation today, please click on the question mark Help widget in the dock at the bottom of your window. Before I introduce today's speaker, I would like to ask everyone in the audience to participate in 4 very brief polling questions. Please click directly on your screen to enter your answers. So here's the first question. Which part of your N-Glycan analysis is the biggest bottleneck? Sample prep, instrument setup, data acquisition, data processing, fluorescence, or data processing MS? So again, click directly on your screen to enter your answers, and that question is, which part of your N-Glycan analysis is the biggest bottleneck? Sample prep, instrument setup, data acquisition, data processing, fluorescence, data processing, MS. All right, fantastic. So let's now go to our second question. So our second question is now on your screen, and it says, how many N-Glycan samples do you process per month on average? Fewer than 10, 11 to 30, 31 to 50, 51 to 100, or more than 100? Again, you can click directly on the screen to enter your answer, and that question is, how many N-Glycan samples do you process per month on average? Fewer than 10, 11 to 30, 31 to 50, 51 to 100, or more than 100? All right, very good. Thank you, everyone. Now let's move to our third question. I'm waiting for that to come up on the screen there. There we go. Okay, so here's that third question. Which aspects of N-Glycan analysis are the most important to you? Sample prep speed and ease of use, LC resolution, data processing speed and ease of use, sensitivity, reproducibility, reliable quantitation. So again, which aspects of N-Glycan analysis are most important to you? Sample prep speed and ease of use, LC resolution, data processing speed and ease of use, sensitivity, reproducibility, reliable quantitation. Give you just another moment there to answer that one. Okay, excellent. So let's now move on to our fourth and final question. Okay, so here's our final question. And again, click on your screen to enter your answers. And here's that question. Do you currently incorporate glycoprotein and glycan standards into your glycan analysis workflow? And if so, where do you source them from? So yes, we use an in-house standard. Yes, we obtain standards from the same vendor as our glycan sample prep supplies. Yes, and we obtain the standards from a different vendor from our glycan sample prep supplies. No, we don't include standards in our workflow. or we're not currently doing glycan analysis. So once again, that question is, do you currently incorporate glycoprotein and glycan standards into your glycan analysis workflow? And if you do, where do you source them? Yes, we use an in-house standard. Yes, we obtain standards from the same vendor as our glycan sample prep supplies. Yes, we use standards and we obtain them from a different vendor from our glycan sample prep supplies. No, we don't include standards in our workflow, or we're not currently doing any glycan analysis. All right. Fantastic. Thank you, everyone, for taking our 4 polling questions. It's now time to introduce today's speaker. We're very pleased to be joined today by Dr. Aled Jones. Aled Jones is a Senior Product and Applications Manager at Prozyme, focusing on tools for glycobiology. He received a bachelor's degree and doctorate in biochemistry from Cardiff University, completing graduate work in the Connective Tissue Biology Laboratories. That was followed by a postdoctoral fellowship at Wyeth Research in Cambridge, Massachusetts, where he subsequently worked as a senior research scientist. Aled, thank you for joining us today. Please get us started.
Thanks very much, Laura, for the introduction, and thanks everyone for joining us today. I'll give a brief outline of what I'm going to discuss today. So first I'll cover the importance of N-Glycan analysis for biotherapeutic development, and then I'll get into choosing a dye for release glycan analysis workflows. And there are 4 dyes that I'll cover in this talk. So Instant PC, which is our latest dye for HILIC separation, which has high fluorescence and mass spec signal. 2AB, which is well known to many for HILIC. Instant Q, which is our dye for screening by capillary electrophoresis using the GlyQ system. And finally, APTS, also for CE. I'll also get into some detail around HILIC separations for labeled N-Glycans with fluorescence and mass spec detection. And I'll include a case study on rituximab biosimilar development with a collaborator that uses 2 of these dyes, Instant PC and Instant Q. Before I get into the main part of the talk, I'd like to emphasize that we'd like to hear from you if you have any interest in the other areas that Prozyme work in that aren't the primary focus of this talk, and the email is there at the bottom. So although glycomics has not had the same attention as genomics and proteomics, around 1/5 of all proteins in the SwissProt database are known to be glycosylated, and glycosylation can impact protein folding, stability, and function. Glycosylation imparts diversity based on differences in glycosylation site, composition, and glycan length and structure. As it relates to biotherapeutics in particular, more than 60% of marketed biotherapeutics are glycosylated. And the 2 main types of glycosylation seen are the addition of O-linked glycans and N-linked glycans. N-linked glycans, where glycans are attached to the amide nitrogen of the asparagine residue within a defined consensus sequence, is the glycosylation type that is most often seen in biotherapeutics. So monoclonal antibodies, or MABs, which are based on recombinantly expressed IgG, are the largest class of N-glycosylated biotherapeutics. And there's a single N-glycan site at asparagine 297 within the FC region. And this slide shows glycosylation in the context of other post-translational modifications, or PTMs, that may be assessed during biotherapeutic development. It's worth noting that there may be additional N-glycan sites in FC fusion biotherapeutics if the fusion partner contains them, and in some cases, additional glycosylation sites even on MABs in the FAB region. So for FC fusions, this is the case for Enbrel, which I'll show a few data slides on later. The generic name is etanercept. And as far as the therapeutic effects of these N-glycans, this may vary depending on the molecule, but these are perhaps the best known as it relates to IgG molecules. So going from the non-reducing terminal of the glycan on the left, sialic acid is known to impact clearance and may or may not be anti-inflammatory depending on the linkage. Galactosylation, the yellow circle residue there, can affect CDC. And core fucose, the yellow triangle, can affect ADCC activity. So there's been efforts in the last few years to reduce core fucose on N-glycans to affect ADCC activity. High mannose N-glycans, such as MAN5, shown at the bottom of the slide here, are immature N-glycan structures, may be indicative of a suboptimal cell culture process. So may be seen in higher amounts if cells are in a state of stress, for example, from being driven towards a higher titer. And the presence of high mannose N-glycans, such as MAN5, can increase clearance. So this is a structure that scientists are monitoring when developing biotherapeutics. As many therapeutics are made in non-human expression systems, non-human glycans are also monitored due to potential immunogenic effects. So humans lack the ability to make the gal, alpha-gal, or galili antigen, and this glycan moiety has been associated with adverse immunological responses when present on glycans of the Fab region of rituximab. Interestingly, the effect is not observed when the alpha-gal moiety is present on Fc glycans, such as in abatacept. And finally, the non-human sialic acid NGNA, or NuGC, is also monitored due to potential immunogenic effects. So knowing the glycan structures that are present is part of understanding biotherapeutic, particularly if glycosylation is known to be a critical quality attribute, or CQA. But how do you go about analyzing what glycan species are there? This slide shows that there are a number of techniques available, each with their own advantages and drawbacks. So going from the top down, intact mass analysis with high resolution, accurate mass spectrometry after reduction or separation of the protein into domains enzymatically can start to give you an idea of the glycan species present, although it may not be possible to get completely down to a granular level on all the species that are there. At one level down, digestion of the proteins into peptides and glycopeptides followed by mass spec, often coupled with a database, is useful for giving site-specific glycosylation information. And for the purposes of this talk, I'll be focusing on released N-glycan analysis, where N-glycans are released with an enzyme such as PNGase F, after which they're labeled to enable fluorescence detection after LC or CE separation. As unlabeled glycans are tough to detect with UV or fluorescence. And the data output for this analysis is relative percent area of N-glycan species. And finally, there at the bottom, it's possible to fragment glycans into their individual monosaccharide components, and then quantify each monosaccharide. For released N-glycan analysis, the sample prep for this has traditionally been a bottleneck in the past. And in the past 6 to 7 years or so, the time taken for sample prep has decreased significantly based on a number of solutions available on the market. So going from the bottom, if you're running a traditional deglycosylation, possibly overnight, and using traditional reductive amination dyes to label your glycans, this can take 1 to 2 days. The GlycoPrep platform that Prozyme launched around 6 or 7 years ago takes 4 to 5 hours with reductive amination dyes like 2AB or APTS, and 3 to 4 hours with instant dyes. And then in the past couple of years, we've shortened this to 2 to 3 hours for 2AB and less than 1 hour with instant dyes. So for the 1 hour sample prep that is our latest, what does this look like? Well, it's powered by a 5-minute deglycosylation, which is enabled by a 3-minute denaturation that opens up the glycoprotein. And this rapid deglycosylation leaves N-glycans in their glycosylamine state, which can be labeled with instant dyes, such as instant PC and instant Q. And reductive amination labeling with traditional dyes, more traditional dyes, such as 2AB or APTS, may also be used. And for all preps, the excess free dye is cleaned up by a vacuum filter plate, and labeled glycans are eluted, ready for separation by LC or CE, with fluorescence detection and the option to hyphenate to mass spec also. So for the 5-minute deglycosylation, we've seen that this is achieved for most of the targets we've looked at. And here's one example of the deglycosylation with rituximab, shown on the right of the slide there, where there are two N-Glycan sites, one in the FC domain and one in the Fab domain. And the blue bioanalyzer trace shows the intact molecule, and the red trace is with PNGase F digestion, and it's shifted to the left with deglycosylation. The light chain is not glycosylated, so remains unchanged in this case. It's worth noting. So there are a number of labeling choices once you have your released glycans, either with instant labeling, shown in the top half of this slide, or reductive amination in the bottom half. So for instant dyes, we have instant PC, which is our latest for LC with fluorescence detection and mass spec, has a strong fluorescence and mass spec signal. Instant Q for CE on the GlyQ system, and I'll be going into more detail on that later, and also instant AB, which is our first generation instant dye for LC. Then at the bottom there, for reductive amination, the glycosylamine form of the glycan undergoes accelerated degradation to the free reducing N form with a hydroxyl at the end, which is suitable for labeling with dyes such as 2AB, APTS, or 2AA. I don't show procainamide here, but that's also a reductive amination option. So 2AB, the label makes the N-Glycan less polar, and it's being used in helic separation of N-Glycans for many years and is well established. APTS has 3 negative charges for CE, so that neutral glycans may migrate with CE, and obviously it makes the N-Glycan more polar. And then 2AA I show here because although we don't offer labeling or workflows for it, we do have an extensive range of standards for 2AA. So first I'll focus on instant PC, and a little bit more background on this. Stands for instant procaine, and here's the structure here. The tertiary amine gives a high signal in mass spec in positive mode, and the activated NHS carbonate ester provides reactivity with glycosylamine groups, and this labeling of N-Glycans is achieved at 1 minute at 50 degrees. Looking at the fluorescence and mass spec properties of instant PC versus a few other available dyes, it has greater fluorescence than any dye we tested, and also has strong signal in MS in positive mode. And this is looking at a single peak G0F. So once you have your labeled N-Glycans labeled with any number of the dyes, depending on your choice, what do you do with them? So the most popular technique is LC separation using hydrophilic interaction, liquid chromatography, or helic. Glycans separated by helic are then detected by fluorescence, and then you calculate the relative percent area of each peak. And MS hyphenation is an option to get towards glycan ID. Shown on the right there is a example of a 60-minute helic gradient starting at high organic, then eluting the glycans with low organic as the method progresses. And these gradients and methods can be adjusted depending on the molecule and the glycan profile that you see from that molecule. There are a number of choices available for helic columns. I haven't included all of them here, but these include the Agilent Advanced Bio Glycan Mapping column and the Waters BH glycan column. So what does the separation of labeled N-Glycans from Biotherapeutics look like? Here are examples for a typical IgG produced in CHO, MabThera, and an FC fusion NBRO. And rituximab is mostly neutral fucosylated bioantenary glycans with a low amount of sialylated glycans and some amount of MAN5. Etanercept has more sialylated glycans on the bottom there due to sialylated species on the receptor portion of the molecule. And I should note that this is a helic injection of 1 microliter equivalent of glycans from 0.4 micrograms of protein. So this is injection of the eluent from our preps. So zooming in on etanercept or Enbrel a little here. So the table on the right is an example of data output, relative percent area of 3 preps. You can see that these CVs are pretty low. They may climb higher for low abundance glycans like A1F6, for example, but that's to be expected with the lower abundance glycans. For the higher abundance glycans, CVs are low. So with any newer dye, like the instant PC separations that I've shown here, there is comfort in having glycan standards available. And these are a list of the instant PC labeled N-Glycan standards that we have available. We also have a CHO-MAB glycoprotein standard and the associated N-Glycan library, which is now available. And you can read more about these standards on our website. But really what we're trying to do here is to cover most of the species that you would see on a monoclonal produced in CHO. So we have alpha-2,3 sialylated glycans as well as alpha-2,6 sialylated glycans that you might see on human IgG or from human cell-based expression systems. So here's an example of a situation where you might want to use a standard. And it's where you have isomeric glycans of the same mass and basic structure, but with different sialic acid linkages, such as this example, A2F or G2FS2, which is biantenary fucosylated sialylated glycan. So in CHO cells, the sialic acid is alpha-2,3 linked. And as I mentioned, in human IgG, the sialic acid is alpha-2,6 linked, and retention time of the alpha-2,3 form is slightly shorter, as seen by the separation by Hillock of these two standards, the alpha-2,3 and the alpha-2,6 form. Enzymatic treatment can also be used to selectively remove sialic acid to determine linkage also. And here's a couple more of the examples of where instant PC labeled N-Glycan standards could be used. If you're looking at IgGs made in CHO, they'll mostly have G0F, G1F, G2F, and a small amount of MAN5, and this is reflected in our CHO-MAB standard shown here. There's also potentially a small amount of G0 minus N and G0F minus N, these smaller glycans that appear at the beginning of the profile. And then on the bottom there is human IgG. Does have a different repertoire of N-Glycan species, so it may not always be the best standard for a CHO-MAB analysis due to these differences, such as sialic acid linkage, the presence of bisecting glucanac species, and also a low amount of high mannose species. We also have an instant PC glucose homopolymer ladder. So this may be used to generate glucose unit values for sample peaks, which is another way of assessing unknown peaks within your sample. In addition to using GU values, you can also use mass spec, which is another step on from standards. And using a dye with high mass spec signals, such as instant PC, you can pretty much see your fluorescence profile mirrored in the base peak chromatogram, which is useful as you hunt for glycans. So in this example from MabThera, you can see MAN5, which is eluting after the main peak G0F, and shown at the bottom there is the mass spectrum for MAN5. And with instant PC in positive mode MS, most biantenary instant PC and glycans will give these known isotopes. So I'm not going to go too much into mass spec. We have some tech notes that address this a little further on our website. So that's instant PC, which, as I said, is really our latest and greatest N-Glycan dye. I did mention 2AB and reductive amination earlier. So why would you want to use this? Well, it is well established. It's been used for more than 20 years. It does have a lower fluorescence and mass spec signal than instant PC, but we found that people may need to match historic data that they've generated using 2AB. So that's why we make this available, and it can be a useful tool in the toolbox. So similar to the instant PC prep, this workflow relies on the denaturation up front to open up the glycoprotein that allows for the 5-minute deglycosylation. And then the new thing here is the lack of dry down before 2AB labeling. So the glycans are passed over a cleanup plate, and the glycans are then labeled on this cleanup plate. The free 2AB is cleaned up on that same cleanup plate, and labeled N-Glycans are eluted in water. And the workflow time is about 2 hours as compares to 1 hour for the instant PC prep, but still considerably quicker than older 2AB preps. So here's what MabThera 2AB N-Glycans look like. And again, TVs are pretty low, shown in the data table on the right there. And again, this is a 60-minute helic separation on the advanced bioglycan mapping column. And a frequently asked question when people look at different dyes, particularly comparing the older dye such as 2AB to the newer dye like instant PC, is how do these match up? Well, the elution order is the same, but 2AB has a shorter retention time than instant PC. So you don't have to stop at one helic method. I've been showing examples of 60-minute helic methods, but in this case, you can actually see improved resolution if you go to a shorter method using a different column. So the top shows MabThera N-Glycans on a 2.1, 1.8 micron column, and the bottom trace is a shorter method on a 2.7 micron column, with slightly improved separation of MAN5. And similar to instant PC, we have a wide array of 2AB N-Glycan standards and libraries to support 2AB labeling of N-Glycans. So I'd like to show the instant PC label in a case study looking at N-Glycan screening and characterization in rituximab biosimilar production. This was done in collaboration with Atos, who are located pretty near to Prozine in the East Bay. So for this work, instant PC was used for LCMS, and the negatively charged instant Q dye was used with our Glycosilation system for N-Glycan screening. So some more background on screening with CE now. So the idea for the Glycosilation system is that you can obtain processed N-Glycan data for 96 samples within a single workday, so 8 hours. And this is achieved by using our sample prep kits in conjunction with labeled standards, custom software that we built for the purpose, and the Glyco instrument. So the Glyco instrument is a pretty small footprint, capillary electrophoresis core with LED-induced fluorescence rather than laser-induced fluorescence. And it runs using this Glyco cartridge, which is a pen shape and is easily replaceable, so it doesn't require any cutting of capillaries. And this cartridge enables a 2-minute separation of each N-Glycan sample. And it's this 2-minute separation which really drives the ability to obtain all this data within a single day. So here's how we ease up the data processing with Glyco. With every sample, upper and lower migration standards are co-injected. So on the lower end, shown at the top left here, DP3, which is a glucose homopolymer of 3 units with a fluorescent label, and then on the higher end, DP15, which is 15 units. So co-injection of these migration standards and comparison to a ladder shown at the top right, which is injected at the start of the sequence, allows for sample alignment using our software, and also the generation of GU values. So you can see the sample alignment at the bottom left, and this is achieved using the migration standards. So once you've obtained these GU values using this method, they can be used to interrogate a GU library of instant Q labeled N-Glycan standards to help you assign peaks. So this is the current state of our N-Glycan GU library for the GlyQ instrument, 55 structures, and they're grouped here by neutral, sialated, and high mannose, and again provides pretty good coverage for most N-Glycan species that you would see in, for example, monoclonals. Also contains alpha-2,6 and alpha-2,3 sialated N-Glycan GU values, which can actually be separated by the system. And similarly to instant PC and 2AB, you don't have to rely on GU values. You can also use standards to support your identification work. So for this case study, we looked at high throughput biosimilar candidate clone screening, and due to the prohibitive cost and time required for N-Glycan analysis, currently a lot of workflows only allow for a few clones to be screened at a time in terms of looking at the N-Glycan profiles. So in this work, the rapid screening that we can do enables 400 clones to be easily screened based on the titer and top candidates selected based on N-Glycan analysis also. So N-Glycan analysis was performed earlier in the process than it would be normally, and also at the 25 clone stage, and also after the top 10 clones had been selected. And then after these top 10 clones had been selected, we also looked at the N-Glycan profile using an orthogonal method, GlyX with instant PC and separation by helic. But going back to the GlyQ separation, this is capillary electrophoresis, or CE, so it's a little different to a helic separation. The glycans are separated first by their negative charge, by that characteristic, and secondly by their hydrodynamic radius. So that's why you see the sialated glycans come out first, followed by the smaller to larger neutral glycans. So shown here on the top there is the innovator molecule, so rituximab, and again, mainly G0F, G1F, with a small amount of MAN5, and at the bottom, the rituximab biosimilar. So this is a pretty busy slide, but it basically summarizes the N-Glycan work that was done during clone screening, and that it can be done, and that this clone screening can be done based not only on titer, but also on N-Glycan profile. So figures 4A to C show relative percent areas as reported with the GlyQ instrument, and figures 4D to F show titers from the screening of example samples during the cell line development and process development, leading to this creation of a cell line capable of producing a biosimilar equivalent to rituximab. So 4A shows the screening of top expressing pool, where each pool differs in the applied expression technology, and 4B shows the profiles of exemplary high titer clones at the batch quantification stage. And 4C shows N-Glycan profiles one step down at the fed batch quant stage. Moving on, this was looking at process development once these clones had been selected. So upon selection of several top clones from the fed batch quant stage, the first set of process optimization experiments were started. So data labeled 1 to 9 is a subset from different process conditions from an optimization experiment. And the bar chart shows the N-Glycan profile, so the relative percent area of each N-Glycan species as compared back to the blue bar, which is the innovator molecule, MabThera. And from this work, it was determined that the N-Glycan profile from condition 1 with the star there on the right was closest to that of the MabThera innovator based on the GlyQ data output. And the N-Glycan profile of condition 1 was further tested and confirmed by UHPLC HILIC, which is shown here. So we have the ATOS sample, which is the biosimilar in red, and the reference standard in grey. Relative percent areas match up quite nicely. So this orthogonal method allows for additional confidence in confirming your N-Glycan profile. And switching dyes to instant PC and analysis method to HILIC also allows for mass spec detection for additional confirmation of the structures that were present. So this work continued, and the biosimilar candidate and optimal process conditions established at shake flask level was scaled up into 3-liter bioreactors, and again the N-glycans were profiled at this point also. So the scale-up was replicated 5 separate times, and the N-glycans from the harvested material was analyzed using instant PC and UHPLC analysis, and the error bars plus or minus 1/7 deviation. And the ATOS process was compared to 4 lots of the rituximab innovator molecule and was deemed to be within 1.5 standard deviations of the innovator product for all of the major glycan species. And this 1.5 standard deviation criteria was what ATOS were looking for here in their work. So using the GlyQ prep, which is 1 hour, and the GlyQ screening, which is 2 minutes, greatly reduced the labor for N-glycan sample prep and data analysis per sample, so allowed for screening during clone selection and process development. And this enabled a significant increase in N-glycan sample throughput due to the decreased runtime and prep time as opposed to conventional methods such as UHPLC. Although that's not to say that UHPLC HILIC can be used later on in the process as a confirmatory method. So if you can do N-glycan work at this early stage, it allows early identification of high-expressing candidates not only based on titer but also the CQAs such as N-Glycosylation. And this reduces risk and enables timeline optimization by early rejection of clones that don't meet your CQA criteria, which in turn enables faster development timelines, so less than 4 months from transfection to selection of top clones. So I'd just like to get into a sidebar at the end here. So one other way to work towards glycan ID, and I've talked about mass spec and standards, is the use of exoglycosidases, which remove terminal monosaccharides from the glycan based on the specificity. For example, sialidase removes sialic acid. So this has been in the literature for a good while, but this is our take on it, which is to use the exoglycosidases on the intact protein, and we have a tech note that's about to be published on this. So use the GlyX protocol, so denaturation up front, but the exoglycosidases are added at this point before deglycosylation, and then these glycans are subsequently labeled and cleaned up and eluted for further separation. So here's an example of this exoglycosidase protocol using the GlyQ system. So the bottom green traces intact N-glycans from MabThera, so all the glycan species are present here. And then moving up, first there's the digestion with sialidase only, and then up from there sialidase and galactosidase, then finally sialidase plus galactosidase plus hexosaminidase, which will collapse biantennary glycans, MAN3 and MAN3F, and leaving high mannose peaks where they are. So this can be an additional method to work towards glycan ID. So that's what it looks like for CE. Here's an example for HILIC. So the top green trace here, in this case, is N-Brel N-glycans, so more sialated glycans here. Using sialidase A, you can see that those sialated glycans collapse. And then adding beta-galactosidase removes G1F, G2F, G2, and then adding N-acetyl hexosaminidase to that collapses everything, as I said, to MAN3 and MAN3F, with a small amount of MAN5 left out there. And finally, I've talked about instant PC, instant Q, and 2AB, but I'd like to mention that we do have a workflow for APTS, which is a negatively charged dye that has been used for some years now for capillary electrophoresis, and this is to support people who are still using the APTS dye in their workflows. Now, we had an existing kit for this, which was Glycoprep with APTS, but we added the APTS Express with GlyX kit to reduce the sample prep time to around 2 to 2.5 hours. And we utilized the no dry down concept that we used for 2AB. So APTS glycans can be separated by CE, for example, on the PA800 Plus, shown in panel B there, and also on our GlyQ system. APTS labeled N-Glycans can be also separated by Hillock, but the peak shape isn't as optimal as it is for dyes that are geared towards LC, like Instant PC and 2AB. And then similarly to Instant PC, Instant Q, and 2AB, we have a range of APTS labeled N-Glycan standards and libraries that can be added to your workflow to support it. So I'm coming to the end of my talk, and in summary, the sample prep time for N-Glycans has really been speeded up over the last few years. It's using the same kind of chemistry, so the same enzymatic digestion, but everything kind of ramped up. So I've covered the choice of dyes that you can use. Instant PC, 2AB, Instant Q, and APTS, depending on your separation and your analysis needs. Maybe you need to compare back to past data. And I've also shown the GlyQC system. So this is a fast separation with easier data analysis based on our alignment of samples. And I should point out that you can also export data from here to using your chromatography package of choice. And this is suited to applications like I showed for the N-Glycan profiling of large numbers of samples, such as from biotherapeutic cell line development. So with that, I'd like to acknowledge the contributions of my colleagues at Prozyme, who are contributing to this work and ran experiments also, and also our collaborators at Atos Biologics. And I'd like to thank you all for attending, and for Laura for hosting this webinar. And with that, we can move on to any questions.
Thank you very much, Aled. It is indeed time now for the questions from the audience. As a reminder to all of you in the audience, if you would like to submit a question, you can do that by typing it in the Q&A box, which you can find on the right-hand side of the presentation window. Okay, so let's get started with the first questions. So here's one, Aled. Could we use nanotechnology for the same purpose?
I'm not sure quite what is meant by nanotechnology in terms of N-Glycan analysis, at least. So that's something that I probably have to get back to the questioner on offline.
Okay, great, thanks. Here's another one. How much glycoprotein can I use with a GlyX sample prep platform?
We suggest using 40 micrograms per prep, but you can go up to 100 micrograms for monoclonals like rituximab. For the lower limits, it may depend on the sensitivity of your instrumentation, and also how low you want to go in terms of looking at low abundance glycans. But we suggest anywhere from 1 to 40 micrograms as a good starting point.
Okay, this question's a little long, so I can repeat any of it if you need me to. Is there a label that would improve separation or retention on a wax column? APTS does add more acidity, but the concern is with poor peak shapes in LC. Is there something similar to 2AA that might work?
It's a good question. You know, the data that I've seen is also for 2AA as being, you know, pretty optimal for the wax or even the mixed mode columns that are out there. So yeah, as far as I know, 2AA is good, as is 2AB.
Great. Are these N-Glycan prep protocols automatable?
Yes, they are. So we have a tech note showing automation of the instant PC and instant Q preps on Hamilton instrumentation, and we're also looking at other platforms as well.
How many samples can I process with the kit?
They come in two sizes generally, so 24 count and 96 count. And you have the choice with those kits of either doing one sample at a time or 24 or 96 samples at a time. You know, we wanted to make them flexible enough so that if you have pretty low throughput and only need to run one or two samples, that we provide enough reagents for you to do that and not limit you to a minimum of 8 per time, for example.
Can I store the dye and use it later? And what about the denaturant?
Yeah, the denaturant is stable at 4 degrees. The reconstituted dye can also be resealed and stored at -20. And for instant PC, this is up to 3 months and 10 freeze-thaw cycles. So again, we wanted to make sure that that people could get the most out of these kits.
How do I store the cleanup plate after use?
Room temperature, and actually we provide caps that show which wells you've used. So we supply the plate all with white caps, and then once you use a well, you can cover it with a black cap, which as well as letting you know that you've used the well, also preserves the seal so that the vacuum plate operates as intended.
Until now, labeling has required purified or captured material. Do you have any thoughts or ideas about using cell supernatant samples for labeling, thus avoiding protein A purification steps, or the step?
Yeah, this is something we've looked at. Unfortunately, if you do your N-Glycan prep on a supernatant, then you're also going to pick up the N-Glycans of whatever other glycoproteins are in your supernatant, in addition to presumably your target that you're trying to look at with a CHO cell expression system or other cell types. So this can affect looking at lower abundance glycans and also signal in general. So it's definitely an interesting concept, but at the moment it's still cleaner to purify the target first with, for example, protein A if it's an FC containing molecule before N-Glycan release and labeling.
What is the most common adduct seen in MS analysis of instant PC labeled glycans?
So this was in one of my slides. Let me get back to it.
Yep.
Yeah, so it is.
And you know you can push that out to the audience, that slide, once you find it.
Okay, thanks. So yeah, in positive mode MS, most biantennary N-Glycans are going to be M+2H, so that's the 2+, and then larger sialylated glycans are going to be majority M+3H.
Right, and did you want to push that slide out? If you hover it over, you'll get a little message that says you can push it out. Okay, great, thanks. Here's another question. I don't have the vacuum manifold suggested in your protocol. Will it work with other manifolds?
Yes, we advise you just to contact us before you get started, and we can provide some guidance.
Fantastic. Well, with that, I think we should wrap up. Alan, thank you so much for your talk today and for answering all those questions.
No problem.
I also want to thank everyone in the audience for attending today and participating in our event, including all of our polling questions. And I'd like to thank our sponsor, Prozyme, for making today's webcast possible. You'll receive an email from LCGC and Biopharm International alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.